Okay, welcome to class. So we are beginning Module 10 of the Apologia General Science curriculum that we've been using all along. And what I need you to do before we even get started is go ahead and grab a sheet of paper. So if you don't have a paper out already, I need you to do that. And I'm going to be asking you questions, and since we're not in a live classroom setting, when I ask you these questions, I want you to be able to jot down your answers so that you can compare what I then tell you and make sure that we're on the same page. And if you feel that you're learning some of this information for the very first time, then you need to go back over some of the previous modules and get a grasp. Make sure you have a grasp on that material before we continue to move forward, and especially with Module 9, because Module 9 is the catalyst for the remainder of the book. So if you don't have a piece of paper already, grab it, and as I ask you questions, you want to go ahead and write them down. So we learned with Module 9 that there were certain criteria. There was a system that was used to be able to determine whether an organism was considered living. Okay, so for instance, you learned that a rock was non-living, but a plant was living. And what was some of that criteria that was used? Do you remember? So I want you to right now write down what the criteria is that we used to determine whether or not something was living. There were 4 criteria that were used. So go ahead and write your answers down. And you can pause this video while you are thinking about your answers, and then you can come back and listen to what the answers were. So you should have written down, does it contain DNA? So for instance, if I were going to look at this book and I wanted to determine whether or not this book had or is a living organism, some of the questions I'm going to ask are, does it contain DNA? Can it grab energy from its environment and transfer energy to the environment? Can it respond to changes in its environment? And can this book reproduce? If you answered yes to any of those questions, then this is a living organism. But if you answered no, it's not a living organism. Alright, so scientists will classify things according to whether or not it is living or non-living, and that's a huge, big scope of classification. So what Module 10 is going to do, it's going to introduce you to an idea that we're going to classify living organisms even further, break them down even a little bit further. So what does that really mean? Well, we are going to look at just another way to take that broad category of something living and trying to make it a little bit more manageable. Alright? Alright. So this classification system that we're going to use is actually for called the kingdom system of classification. It's a kingdom system of classification, and there are 5 kingdoms that we're going to talk about. So in this kingdom, in this classification system of kingdoms, keep in your mind that everything here are living organisms. So if they're all living organisms, and we're going to break them down into 5 categories, well, how are we going to see, what are we going to use to determine that difference between each of those living organisms? So what we're going to look at is actually at the cellular level. Excuse me. At the cellular level. And what we come up with is characteristics that are similar at the cellular level and characteristics that are going to be different, and that's why we're going to be able to classify organisms into different groups. All right? So for now, I want you to write on your paper that there's a 5-system classification system known as kingdoms. a 5-system classification. I see I'm saying system again, so you can word it in your own words, but basically it's a class system that equals 5 kingdoms for living organisms. A class system that equals 5 kingdoms for living organisms. And those kingdoms are Monera, Protista, Fungi, Animalia, and Plantae. And your book has Plantae and then Animalia. Don't be confused with the last two because you know that they look like plants and animals, and they are. We're just using the Latin words here, Plantae and Animalia. So the 5 systems again are Monera, Protista, Fungi, Plantae, Animalia. Now, the last module, in Module 9, you were introduced to 3 types of cells. On your paper, I want you to write 3 types of cells that you were introduced to in the last module. What were they? Okay, you should have written down plant cells, animal cells, and cells without a nucleus. So that's where we're going to begin. Cells without a nucleus. Now, I'm not sure in your textbook, since you have edition 2 and I'm using edition 1, the actual figure, but your textbook in figure 10.1 for me does a nice job of showing you the difference between prokaryotic and eukaryotic cells. And I'm holding this uneven. There we go. So you can see the sketch of a prokaryotic cell has a strand of DNA. That DNA, you know from our last module, contains all of the genetic information that that particular cell needs to be able to multiply. All right, it has all of the genetic information that makes that cell that cell. The eukaryotic cell also has DNA, but it's housed inside the nucleus. And if you look at the eukaryotic cell, you can see all the other organelles as well that are in there. And some of those organelles should not be unfamiliar to you if you played the plant cell game. All right, they're going to be very similar as what you see here. So with that plant cell game, you really do want to make sure that you know the structure, like what is inside that cell, and then the responsibility for that structure, what it actually does inside the cell, and then you want to know where it is located. We're going to do that for plant and animal cells, so I'm going to send you the link this week for the animal cell. By the end of this week, you're going to be able to tell me the difference between components that are in the animal cell versus components that are in the plant cell. Okay, so remember that the 3 different types of cells: plant cells, animal cells, and then cells without a nucleus. Alright, that brings us to Kingdom Monera. Kingdom Monera. Let me just make sure I'm correct with my notes here. Hold on. I'm sorry. Cells without a nucleus are prokaryotic cells, and Kingdom Monera does have prokaryotic cells. And Kingdom Monera is unicellular, meaning one cell, and it is prokaryotic, meaning no nucleus, but it does have DNA, so it's going to be able to replicate itself. Kingdom Protista, so I'm going to introduce these and then I'm going to break them down even further later, so I'm just going to give you the 5 right now. Kingdom Protista is a eukaryotic cell, and organisms in this kingdom generally contain algae, and algae are just plants that you find, not really plants, and we'll go over that in a second. They look like plants. They look like water plants. Okay, but they're not really plants, because when you think of a plant, I want you to jot down on your paper, what are some of the things or characteristics that a plant has? What are some of the characteristics that a plant has? Look in the corner over there behind my messy table. I'll go this way, you don't have to see the messy table. No, just joking. But look over there behind the plant over there, and you can see a stem, you can see leaves, and then what's something you really can't see? You can't see the roots. So characteristic of all of plants would be that there's stems, there's roots, and there are leaves. Okay, but algae look like water plants, but they're really not water plants. All right. Later on, you're going to learn that each of those components, and I don't know if it's going to be in general science or if it's when we touch base in biology, but each of those components, the stem, the leaf, the roots, they all have a specific role to play in that plant being able to produce photosynthesis. And I think I just gave you an answer to maybe a question I was going to ask you next, but let me see. So although most protista are microscopic, not all is, because seaweed is a water plant that you can see with the naked eye. So that means that you can see it and you don't need a microscope to be able to see that. The next kingdom is kingdom fungi. Now, kingdom fungi is feeds on dead organisms, and most fungi can be seen with the naked eye, so you don't need to use a microscope, and they are composed of trillions of cells. The most common fungi that is out there is yeast. All right, so yeast is a member of the fungi family, and it is unicellular. It's a single-celled organism. So what you want to remember is that yeast is single-celled. Yeast is a single-celled fungi. Mushrooms are multicellular fungi, but together, or they are both eukaryotic cells. They are both eukaryotic cells. So what does that mean? With yeast, all right, I gotta come back over here. With yeast, I'm gonna see all, this is really hard to do backwards, of these components, okay? I'm gonna have all the components of a cell, all right, that a multicellular organism is gonna have. But yeast, being only one cell big, is gonna have all of these components in it. All right. So on your paper right now, what I want you to do is define what a eukaryotic cell is. I've shown you a diagram and I've told you the difference between the two already. I want you to be able to put that in your own words. So if you need to pause the video, go ahead and pause the video and then come back and we'll discuss a eukaryotic cell. Just waiting for those of you that didn't pause and you're waiting for the answer. Alright, so a eukaryotic cell, you should have been able to tell me, is a cell with organelles. Okay, and remember, all of these organelles, they have their own particular membrane. So a lot of times you'll hear the definition for a eukaryotic cell is a cell with organelles that contain their own membranes, or a cell that has DNA inside a nucleus. So they sort of go hand in hand, okay? But you want to be able to know that a eukaryotic cell has DNA inside the nucleus and has membrane-bound organelles. All right. The next kingdom we're going to discuss is kingdom Plantae. Now this kingdom produces its own food. And so we give that name autotrophs, meaning autotrophs. Automatically, they're going to produce their own foods. And they do this by a particular process we call, and I sort of gave you the answer early on, but what is that answer? What is that process? By what process do plants produce food? So go ahead and write that down on your paper. And if you said photosynthesis, you are correct. So remember, we already discussed in the last module that photosynthesis is a chemical process, meaning that it's going to take some stuff in, it's going to transfer it, and it's going to give us some byproducts or some end products. All right? So what are those things that it's going to take in according to module 9? Go ahead and write that down. 3 things you need for photosynthesis to take place. So photosynthesis needs, that's how you should be starting that. Photosynthesis needs carbon dioxide, which you could have abbreviated as CO2. It needs the sun, and it needs water. So are plants prokaryotic or eukaryotic? Are plants prokaryotic or eukaryotic? And then I want you to tell me how you know they are the answer that you gave me. Are they eukaryotic or prokaryotic? So plants are eukaryotic. Plants are eukaryotic because they have DNA inside a nucleus and because they have membrane-bound organelles. You learned about all those organelles last week when you did that game. Okay. So kingdom Animalia. Again, do you think these are prokaryotic or eukaryotic cells? Kingdom Animalia. Okay. Eukaryotic. Again, they're going to have specific membrane-bound organelles. Each of those organelles are going to have a specific job to do with inside that cell. And if you don't understand the difference between prokaryotic and eukaryotic at this point, you need to review the beginning part of this chapter, Module 10. You're going to have to go over and reread that really well. Alright, so I'm going to break down now Kingdom Monera for you. Oh, and so I'm sorry, let me just back up. Kingdom Animalia, you should understand, contains all of the animals that you can possibly think of, from the grasshopper to a giraffe. I mean, everything in between, okay? And Kingdom Animalia not only has land animals, but we're going to be talking about land animals, animals of the air, and animals of the sea. So it encompasses a huge group. So when you get into biology, which bio means living, and ology, study of, so when you get into biology and you have the study of living things, you're going to see that the Kingdom Animalia is going to be broken down so much further, and we'll classify things according to their similar characteristics. We'll be able to put them into groups or whatever. We're not going to get into that much detail right now. We're just going to keep with the 5 system of classification here, is known as kingdoms. But later on you would learn that each of those kingdoms would be broken down further into a phylum and then to a class and so forth and so forth. Alright, but right now we're going to look at Monera, Kingdom Monera. So Kingdom Monera is the largest organisms in this kingdom. It has the largest number of organisms in this kingdom compared to other kingdoms. All right. Monera can withstand extreme temperatures. They have found Monera living in 2,000 degrees Fahrenheit on a nuclear receptor. Monera has been found in freezing temperatures. So they can survive in environments that other kingdoms or other organisms in other kingdoms cannot survive in. They live on our skin, they live in our guts, and electron microscopes nicely pick these guys up. Alright, so what we're going to learn with Monera is bacteria. There are bacteria that belong into the kingdom Monera. And bacteria can contain pathogens. And I say can contain because not all bacteria is going to be harmful for us, and we're going to learn about that later on in the module. But for now, we know that a pathogen, so if you don't know what a pathogen is, go ahead and write the word pathogen on your paper. P-A-T-H-O-G-E-N. Pathogen is an organism that contains disease. An organism that contains disease. So can you think of any organisms that contain disease? Well, every once in a while we hear about E. coli. There was a major fast food chain that was just in the news that had an E. coli breakout because of bacteria. We hear of listeria. Sometimes our lunch meats and stuff, or meat packaging plants, we hear of listeria outbreak. And salmonella. Your book discusses salmonella, and salmonella is another bacteria that is responsible for people getting sick. Salmonella is found in meat and in egg products, and so one of the ways we can kill that bacteria or that pathogen is to make sure that we cook our foods to the appropriate temperature. All right, that's just one way. So salmonella, yeah, I already said that. Okay, so but another method that is used to kill bacteria in foods would be refrigeration and salt, vinegar. Those are all ways that would help to slow the bacteria growth or even the reproduction of bacteria. So let's think about this. The experiment, which I told you guys, well, no, you're going to watch an experiment, and then you're going to be able to see how salt, how vinegar, and how temperature are all going to affect bacteria growth. Again, if you don't understand the outcome of that experiment, then just make sure that you go ahead and read that portion of the textbook that will clearly explain that to you. Salt and refrigeration kill bacteria. Let me see. I just want to see what my notes are. Okay, so food that spoils. Oftentimes, you'll have leftovers for dinner, and then you don't want to leave them out on the counter. You want to be able to throw them into the refrigerator, because throwing them into the refrigerator cools any bacteria that would be on that food, and it will not allow for it to grow, and so it just slows it down. That bacteria is still there, but it's not really growing like it should. And then when you reheat the food properly, it kills that bacteria, and then you can consume it without being sick. All right. Salt reduces the growth and reproduction of bacteria, and that's why meat used to be preserved with fish. Fish and meat used to be preserved with salt, and in doing so, the salt actually added flavor to the meat, so it was really advantageous two ways. One, it preserved it, and then secondly, it gave it flavor. But salt isn't the most ideal way to preserve other foods, because we don't want that transfer of taste. So that's why refrigeration is the best method of preserving food and slowing down the bacteria that grows on food. And so vinegar would be the same exact thing. Vinegar works well. People use it when they're pickling food, but we wouldn't want to put vinegar on all things, because it would transfer a taste and it wouldn't be that good. So remember, even though we're discussing bacteria and the way to slow the bacteria down, we don't want to think that all bacteria is bad. All bacteria is not bad. We need bacteria in our system so that our digestive system will work correctly. We need bacteria on our skin so that we can take care of other organisms that could potentially be harmful to us. So bacteria is actually good for us in certain ways, or certain bacteria is good for us. Okay. So your book does do a nice job about discussing that as well. So let me just come over to here. Yeah, so and then there's other bacteria that we use in the processing of food. So we add certain bacteria to milk, and that bacteria then allows us to make cheese and yogurts and things that people without allergies to dairy can consume. Alright, let's see. Kingdom Protista. Kingdom Protista is neat in that we can look at microscopic organisms that are found in pond water, if you will see it, and we can say that they have very similar animal-like qualities to them, and we're going to call these protozoa. And so protozoa, if we look here in your textbook, sorry, this one is figure 10.3 in this textbook, and I'm sure you're going to have a similar one in your book or whatever. But you can see the amoebas, the euglenias, the parameciums there, okay, plasmodium down there on the bottom, okay. So each one of these organisms have a do not have any real an amoeba does not have really a defined shape. It sort of will just exist like as a blob, all right, and it's constantly changing shape. But there are arms, if you will. These arms, these arms are called pseudopods, all right. Pseudo meaning like, and pods meaning feet or arms here. And those pseudopods move around, and it sort of gives the amoeba its blob type consistency as it's moving around. And it has to repeat this process 7 times before it can move from one place to another. Alright. Then the next one, the euglena, are organisms that are smaller than the amoeba, and they move with a tail which has a flagellum type whip-like action. Alright, and these organisms, the euglenas, do not eat other organisms, but they produce their own food with photosynthesis. So they are not a plant, alright, but they do produce their own food with photosynthesis. And then paramecium, this is in your book, that organism is magnified 350 times so that you're able to see it. And they're the largest of the protozoa. And they have a lot of hairs on the outside of them, okay? And I'm just going to go like this because those hairs are cilia, and those hairs are constantly moving. And that's what allows them to go through the water, is that constant moving of that cilia. And the plasmodium, this is magnified, that's the last one on that page, is magnified 700 times, and these organisms cause the disease known as malaria. And they are usually transmitted through mosquito bites. So those are just some of the organisms that you can find in Kingdom Protista. Other organisms that you can find in Kingdom Protista on the next page, figure 10.4 here, is the algae. Alright, so you have diatoms, you have kelp, and you have the red algae down there. Alright, the diatoms are really neat. With your parents' permission, you should get online and just type in the word diatom, and go to Google Images and look at diatoms. And these are geometrically shaped images. This particular one is magnified 400 times so that you can see this. Diatoms are the most important group of organisms on the planet when it comes to photosynthesis. All right. Algae makes up 75% of all chemical of the photosynthesis that we have. So algae makes up 75%, while trees and shrubs and plants make up 25% of photosynthesis. So, and again, as we know that oxygen is a byproduct of photosynthesis, it's really important for us to see that algae is really, really important for us as humans to be able to consume oxygen as well as for animals. All right. So here with the kelp, here with the kelp, we have no magnification. You can see it with your naked eye. There's no need for a microscope. All right. And then the red algae that we're seeing at the bottom is another form of seaweed, and it's interesting because of its red color. And most organisms that perform photosynthesis are green, but red algae is one exception to that general rule. So it's one way for us to always remember about photosynthesis and it being related to plants is because plants is green, but here we see we can have red algae and it can produce photosynthesis. So that is what makes it unique. Kingdom fungi, I'm not going to go into in too much detail right now. So I think I have, let me just make sure. Yeah, we're going to pick up with kingdom fungi. So you're going to read as far, just up to the kingdom fungi in your in your lesson. But hold on one second, I'm just going to get you something. I'm going to show you, we're going to do an experiment, but it's started here, so hold on. Alright, so right now this is basil, and this has been growing on my counter, and this has been growing on the counter in just this little jar, and you can see the roots in here. Alright, be a good picture here. What we're going to do in class next week is we're actually going to cut off all that you see up here, okay, and we're going to just plant this in potting soil, and then we're going to see if we in fact still have basil come up. Alright, because that would be what we can call vegetation, let me just make sure I have the right terminology for you, vegetative reproduction. Okay, so we're going to actually just take the roots and grow a new basil plant. Alright, so this is already started. Alright, and that would be experiment 10.3. And just one other thing, look how nice that basil looks. I mean, that looks really good, right? Alright, because these plants are standing up and they're not like wilted over or anything like that, because they're standing up, that's because of Tuger pressure. The water is able to come up from the roots into the stems, out to the leaves, giving them so that they can stand up as nice as they are here. Your book is going to talk about this, so at least you'll be familiar with some of that terminology. Alright, so I'm going to email you this video, but you already know that because you will have watched this, so I am going to exit out. You do have you do have instructions on the email that I am sending you. If you have any questions, give me a call. Bye.